Go ahead.
This is our bioinformatics analysis of the MM14 Lynx canadensis. I'm Maddie Folsom. We also had Katie Furick and Bailey Robertson. This is for Dr. Petrick for our genetics lab. Katie, you can go to the next slide. Thank you. Bioinformatics is an interdisciplinary field of science that develops methods and software tools for understanding biological data. It is a computer science concerned with the acquisition, storage, analysis, and dissemination of biological data. This project gives us initial exposure to web-based tools to compare DNA sequences. During this project, we will use bioinformatics to navigate computer science to determine the protein used in our sequence. We will be using the NCBI website to determine the amino acid sequence and the function of our protein. We started by choosing our sequence. Ours was sequence MM14 from our list on Blackboard. We started by trimming off what we called the dirty sequence. This was the sections in our sequence with a large portion of ends. We removed the large strings of ends, and this created our cleaned up sequence that you see here on this slide. We then took the sequence and pasted it into the nucleotide blast on the NCBI website. This pulled up the closest match to our given sequence. The closest matched, as shown below, is the Lynx canadensis actin gamma mRNA. This query line, which is the top line that you see in this sequence, that is our sequence. And the subject line that falls right below it is the line that matches our sequence. So that would be the Lynx canadensis actin gamma mRNA.
Okay, so here we have our matching coding sequence with the 5' untranslated and the 3' untranslated region. So the beginning of this sequence in the white from 1 to 61 before the brown is our 5' untranslated. And then our matching coding sequence is this large portion of brown and then afterwards, this last white part is our 3' untranslated region of our protein. Okay, so what is the protein product of this RNA? So our protein product of this RNA sequence is the actin cytoplasmic 1, which is also known as the actin gamma 1, Lynx canadensis. And these are the amino acids in one letter abbreviation. And here is our list of our matches of our protein sequence. And we are going to be talking about the Ovis aries and the Paromyscus californicus insingis. Okay, so our first protein alignment is the Ovis aries. We also have here our query 1 line, which is our original actin gamma. And then our subject line is where the Ovis aries matches. So we had to make sure that both of those lines matched up as close as possible and that all of the letters were the same. So in the query was the Lynx canadensis, and then the subject was the Ovis aries, and then the Ovis aries is actually a sheep. Our second protein alignment is the Paromyscus californicus and insingis. And again, we have our query line of the Lynx canadensis, and then our subject line is the Paromyscus californicus, and that is a field deer mouse, a California deer mouse. And then Bailey.
Okay, so what we are looking at now is the collapsed version of our fast minimum evolution tree when putting our protein through the process of finding the tree our results ended up with the multiple organisms down here that you can see with 4 leaves. Next slide, please. This would be the expanded version of our fast minimum evolution tree. As you can see down below, there's our Lynx canadensis actin cytoplasmic 1, which is the protein we ended up with, as well as the other result of our search, the Ovis aries or the sheep. And now we're looking at our neighbor joining tree, which is the highlighted multiple organisms that has 19 leaves. And our expanded version of the neighbor joining tree, as well as our protein right down there at the bottom under all of this section. You can see it had other families in this tree. So what is actin gamma 1 or actin cytoplasmic 1? The actin gamma 1 is a cytoplasmic actin found in all cell types. It's encoded by the ACTG1 gene. It's widely expressed in cellular cytoskeletons of many tissues, as well as in adult striated muscle cells. It's localized into Z discs and costamere structures, which are responsible for forced transduction and transmission in muscle cells. Mutations of this ACTG1 gene can be associated with many different problems, such as hearing loss, and it has been assumed to be in some cancers, which has not been properly proved. So don't cite me on that. Next slide.
So this right here is my first article. It was about a mutation in the gamma actin 1 gene that causes autosomal dominant hearing loss. The first article that I found discussed the mutation of our cytoplasmic Y actin 1 gene causing autosomal dominant hearing loss. There are 18 known genes involved in autosomal dominant hearing loss, 36 of which remain to be identified, and 17 of those are implicated in autosomal dominant forms. Autosomal dominant hearing loss shows variation in age, progression rate, and type. For individual families with autosomal dominant hearing loss, it is often possible to suggest one or a few loci or genes involved. The experiment found 4 families with autosomal dominant hearing loss that were found to be linked to overlapping regions of chromosome 17q25 and 2 different DNA locus numbers, DFNA20 and DFNA26. In the article, they then tested a 5th family with linkage to this interval. The hearing loss in this 5th family is similar to hearing loss in loci number DFNA20. The article discusses the gene analysis of the 5th family, which led to the identification of a missense mutation in the actin gamma 1 gene. They then tested this missense mutation on the highly homologous magnesium ATP actin 1 of Dictyostelium discoideum, which showed it has a small but significant functional effect. With all of that being said, the methods they used for the experiment include first isolating the DNA and amplifying the actin gamma 1 gene using a thermocycler under precise cycling conditions. They then purified PCR fragments to do sequence analysis using big dye terminator sequencing. Next, they designed ARMS primers for both the wild type and the mutant reverse primers. The amplification was done under precise conditions. They then analyzed the mutation using the DGMUT command, and for every substitution of isoleucine for threonine at position 278 was displayed on the screen. All in all, the mutation in the gene encoding cytoplasmic Y-actin-1 is involved in dominant sensory neural hearing loss related to the loci numbers DFNA20 and DFNA26. Katie, if you'll switch the slides for me. This first figure that you see on the top left shows a mutation of the gene in a family using a pedigree chart. That top gel run shows the mutant reverse ARMS primer, and that bottom one shows the wild type reverse ARMS primer. This image on the bottom right shows the partial sequence of a controlled individual. I have circled, I don't know if you guys can see it, but right there in red, I've circled where they substitute isoleucine for threonine at the 278 position. And you can see where it would be it shows a heterozygous ACC to ATC transition. Katie, if you'll switch slides. This is my second article. It is actin gamma 1 is a critical regulator of pancreatic ductal adenocarcinoma. This discusses actin gamma 1 being a regulator of pancreatic ductal adenocarcinoma. Actin gamma 1 is a kind of actin isoform that exists in almost all cell types as a component of the cytoskeleton, thus mediating cell viability. This study explored the protein expression of actin gamma 1 in human pancreatic ductal adenocarcinoma and identified its clinical significance on predicting patients' overall survival. They first obtained tumor samples from 149 patients who had undergone a surgical resection and were pathologically confirmed as PDAC without distant metastasis. When I say PDAC, I refer to pancreatic ductal adenocarcinoma. Those tumor samples were then formalin fixed and paraffin embedded for immunohistochemistry staining. Next, the human PDAC cell lines, PANC1 and BXPC3, were cultured and incubated. They also underwent a short hairpin RNA transduction. They then used a technique called Western blotting, where the cells were lysed, centrifuged, membranes were blocked and incubated, secondary antibodies were added, and then incubated again. Through this, they were able to detect the signal that they were needing. They then performed cell proliferation to measure the proliferative capacity of PDAC cells. The transducer cells were seated in a 96 well plate and cultured, where they were then treated with a reagent followed by incubation before determining their absorbance. From the data they gathered, they were able to determine the levels of actin gamma 1 mRNA were significantly higher in PDAC tissues than that in non-tumorous pancreatic tissues. To sum that up, patients with higher actin gamma 1 mRNA levels exhibited poorer disease-free survival. These findings caused them to explore its protein expression patterns. The actin gamma 1 protein is widely expressed in the cytoplasm of both normal pancreatic tissues and PDAC tissues. These results are from the IHC staining, which was the immunohistochemistry staining. But the actin gamma 1 protein exhibited distinct expression levels in different PDAC tissues. Patients with more advanced T stage exhibited significantly higher actin gamma 1 protein levels, indicating its possible involvement in tumor growth. If you're wanting to see this T stage, I'm referring to it shown in figure 1 in the article. They then silenced the actin gamma 1 protein, which significantly inhibited the proliferation capacities of both the PANC1 cells and the BXPC3 cells. Finally, they isolated the subcutaneous xenografts and confirmed that silencing the actin gamma 1 protein can suppress PDAC growth. In conclusion, they were able to see that actin gamma 1 protein can promote PDAC progression, at least partially, by enhancing tumor growth. can move on, Katie. These are my cites from both of my articles. The top one is from the first article of the mutation, and the bottom one is from the critical regulator.
Thank you, Maddie. Now for my two articles. For my first article, I had the knockout of the ACTG1. As we mentioned, actins are involved with cell motility. So they wanted to find and evaluate how cell motility was impacted by the lack of the gamma actin coded by ACTG1. They did this by using the use of human melanoma cells. They wanted to use the use of human melanoma cells because the melanomas have high plasticity and they grow fast, which makes them easier to study on. And we want to know why they were doing this experiment in the first place. And from the introduction, they state, because there is no agreement regarding the purpose of the existence of the two muscle actin, it is of great importance to study the function of beta and gamma actin. And we will learn later that they coincide with each other, which is why they want to study them both at once. And the results proved that the ACTG1 was important in migration capabilities because both actins are present but are in different parts of the cell. Gamma actin was found in the paranuclear area, so it was surrounding the outer edge of the cell, and then the beta actin was just below that. And then they also found that the knockout of the ACTG1 messes with expression and cell migration. And ACTG1 was affected by its distance, velocity, and migration direction. So any mutation of the ACTG1 is going to change the way that the cell moves. Okay, so for my second article, they wanted to find what was the new information was in this experiment supposed to bring to the table when it comes to the gamma actin. They wanted to differentiate the beta and the gamma actin as contributors to hearing loss in mice. So mice were the subject in this case. Auditory brainstem response was used. They used this auditory brainstem response to measure the waveforms collected from the mice. They found that the mutations within the ACTG1 is described as being dominantly progressive, which means that the hearing loss happened at a more progressive rate than the beta. The beta and the gamma actin coincide with each other in a cell. The article states, we found that although cytoplasmic actin is necessary, neither beta actin or gamma actin is required for normal stereocilia development or auditory function in young animals. However, aging mice with beta actin or gamma actin deficient hair cells develop different patterns of progressive hearing loss and distinct pathogenic changes in stereocilia morphology, despite co-localization of the actin isoforms. The mutations can cause progressive hearing loss amongst the actin gamma 1. So that's where the gamma was moving at a more progressive rate than the beta. And in this figure, so for my figure of experiment 2, hearing loss and stereocilia pathology depended on the Y-actin concentration. So we had the level of the DD-SPL within the given weeks of the mice, how old they are. And we have our actin gamma 1 up top here that shows how it's the hearing loss is getting progressively worse. And then this was my figure from experiment 1, where you can see the beta actin here on the left picture. It is kind of dispersed throughout the cell, but then we also see in the second photo, the gamma actin is completely paranuclear around the cell. So you see it right along the outer edges, which could show why it involves cell motility. And here are my citations. The first one is for my first article, and the second one is for my second. Okay.
So my first article was on the delayed embryonic development and impaired cell growth and survival in ACTG1 null mice. So obviously mice were the test subject here, and they were testing against an active ACTG1 gene or a null gene in the mice and how it would affect the development and growth inside the embryo as well as outside, and how they would survive continuing on. The gamma-actin null mice were fully viable during their embryonic development, but most had died within a 48-hour period of birth due to either respiratory failure or as well as cannibalization by their parents. They had no morphogenetic defects identified in the null mice, but they exhibited stunted growth during their embryonic and postnatal stage, as well as delayed cardiac outflow formation that resolved by birth. Using primary mouse embryonic fibroblasts, they confirmed that gamma-actin is not required for cell migration, but the null which most likely contributed to the stunted growth and developmental delays observed in the null embryos. The total amount of actin protein was maintained in the null cells. It suggests that a requirement for the gamma-actin protein is required for cell growth and survival. Sorry. I'm so sorry. I just lost my train of thought. Okay. So through a series of tests using both active gamma and null gamma mice, they have compared the multiple stages in embryo and postnatal, and have determined that a stunted growth and delayed cardiac development in the null mice was the result. They had timed matings established between the ACTG1 heterozygous pairs and resulting null embryos examined. The null embryos were morphologically similar to their wild type counterparts throughout development. However, they were at least 11% smaller in size, as measured by crown rump length from embryonic day, E10.5 to E18.5. The null mice remained smaller in size during the first 3 weeks of the postnatal development and into adulthood. The histological analysis of developing embryo revealed a delay in the development of the cardiac outflow tract in the null embryos. During cardiac development, the proximal portion of the outflow cushions fuse and muscularize, eventually walling the aorta in the left ventricle and completing the formation of the ventricular septum. In wild-type embryos, the aorta was observed to be exclusively in communication with the left ventricle. However, in null embryos, this was not observed until later on in development. Furthermore, in the wild-type embryos, but not in the null embryos, the ventricular septum was fully formed along the entire anterior and posterior axis. The ventricular septum was completely formed by the E18.5